everyone. So in this video, I will be talking about how fungi generally trick plant immunity by producing several effector proteins. So a healthy plant is a host for several phytopathogens, which includes both bacteria as well as the fungus. These phytopathogens generally attack the aerial parts like leaves, stem, flower or fruits, or they can also target the roots. For example, there are several root-borne fungus which attacks the root and enter into the plant system. These microbes generally undergo colonization and lead to symptom development. For example, wilting, damping. Okay, there are several symptoms which are associated with the plant disease, and every microbe gives a specific type of symptoms. Okay, and the symptoms, for example, if there is an attack by a microbe and the plant fails to defend such kind of pathogen, it generally lead to complete uh, damage of the plant. Okay, in this scenario, you can see a healthy plant is getting into a complete damaged form. Okay, so what generally happens that if the plant system is failed to respond, then such kind of situations happen. But now the plants have their own defense responses. For example, these plants generally defend themselves using two layers of immunity. The first, very common, is the pattern triggered immunity, PTI. The another one is the effector triggered immunity. So first we will be talking about pattern triggered immunity. In this case, the plants generally detect danger signals from the microbes. For example, these danger signals are termed as pathogen associated molecular pattern, that is PAMP. Any microbe, for example, any phytopathogen produces compounds termed as PAMP that is detected by the plant system. Once these molecular patterns are detected, there is induction of signaling in the plant system in such a way that there is blockage of the fungal entry, or there is a proper defense in which the fungus or the bacteria fails to attack to the plant system. Okay, for example, a fungus basically consists of two components in their wall, for example, chitin as well as beta glucans. These act as a PAMP. In this case, you can see that the plant system basically consists of pathogen related receptors that is specific to such kind of compounds. Once these compounds are detected by this receptor, there is induction of several uh, signaling in the plant system. For example, there is rapid accumulation of ROS, there is simultaneous cell wall strengthening, as well as there is induction of defense genes which are responsible for defending the plant system by blocking the entry of the fungal phytopathogen. Now, in order to bypass this kind of blocking, or in order to bypass such entry, or in order to establish infection inside the host cell, the fungi releases effectors, and these effectors are basically small secreted proteins which are produced by them. They help the fungi to infect and colonize plant tissues, suppress plant immune responses, as well as they can also alter plant cell signaling and metabolism. Okay, so here is a short example where we are going to see that how the fungal strategy blocks the chitin triggered immunity. So in this case, in the picture, you can see that this is a junction where the fungus is trying to invade the plant system. In this case, first we will see the chitin triggered immunity, how it is basically happening. Okay, so in the plant system, you can see a CERK1 receptor which is present on the membrane. This receptor generally recognizes the chitin, and upon recognition of the chitin segments, it generally triggers the immunity inside the plant cell. For example, we have seen in the previous slide that like generation of ROS, then induction of defense genes like this. Simultaneously, plant produces chitinase enzymes which is involved in cleaving the chitin into a smaller fragments and, in order to block their entry. Okay, so this is a chitin triggered immunity in the plant system. Now basically what happens that a fungus, namely Podosphaera xanthine, has been reported to produce an effector protein which is named as CHBE, that is fungal chitin binding effector. These effector proteins here present in blue binds the small chitin fragments. Okay, what basically happens? This CHBE binds the chitin fragments so that they remain unrecognized by the plant system. Hence, there will be no dimerization of the receptor and there will be no initiation of defense signaling. And finally, there will be successful fungal invasion in the plant system and there will be successful establishment of the infection. Okay, so this is the first example where we have seen that the effector protein is involved in bypassing the immune response given by the plant system. The second scenario is that how the effectors suppress the effector triggered immunity. The first we have seen is the pattern triggered immunity, where the plant recognizes certain pattern which are present on the microbes and generate immunity. In second case, uh, now we will see that how the effectors basically suppress the effector triggered immunity. So in both examples we are going to see. Okay, in this case we will see that, for example, a fungal AVR. AVR is the avirulence gene and plant R is the resistance gene and their interaction. For example, AVR is fungal origin. Okay, so in this case, you can see that the conidia is in junction with the plant cell, and upon infection, AVR protein or AVR is released inside the plant cell. This AVR is the effector. Okay, this AVR is the effector protein which is produced by the fungi. Okay, this effector is generally recognized by the plant R gene, that is a receptor gene, and it leads to hypersensitive response within the plant cell which generate rapid cell death only at the infection site, thereby blocking the pathogen spread. Okay, so in this case, you can see that the effector is suppressed by the plant. Okay, so this is the effector triggered immunity which is generated by the plant system. In order to bypass the mechanism, now what happens that if the AVR gene is mutated, for example, if the AVR gene is mutated and then if the fungi, now if it enters the mutated AVR, then it remains undetected with this resistance gene. Okay, as the effector protein is now mutated and the infection is successful, there will be no detection of this protein by the plant system, and now no defense response will be triggered, and finally the pathogen will successfully infect the plant and the disease will happen. Okay, so this is the second response in which the fungi bypasses the effector triggered immunity. Okay, another example is one where there is hijacking host metabolism. In this case, for example, you can see a fungus named Sclerotinia produces an effector, namely SS pine 1. Okay, so it basically inactivates plant defense mechanism, specifically the polygalacturonase inhibiting protein. Okay, so we will see what is happening here. So plants generally produce this PGIP. This PGIP is generally involved in inhibiting the fungal enzyme, that is polygalacturonase. Okay, these polygalacturonase are the enzymes that degrade pectin in the plant cell wall. Okay, so the fungi basically produces this polygalacturonase enzyme in order to degrade the pectin which is present in the plant cell wall. Okay, now what happens? This S-spine effector protein directly interact with and inactivates this PGPI. Okay, now when the enzyme is blocked, the fungi can produce the polygalacturonase enzyme which can degrade the pectin, and now the fungi will be capable of breaking down the plant cell wall and facilitate its infection, which ultimately leads to uh, tissue maceration as well as disease development. Okay, so this was another short example. Okay, so overall, why this knowledge matters? Generally, it helps in developing disease resistance crop. Then it supports genetic engineering of plants. It also enhances understanding of plant pathogen interaction, and ultimately it will lead to improved crop protection strategies. So thank you for watching. If there is any query, or there is if there is any additional information related to it, you can mention in the comment section. Thank you so much.